Protein analysis is a high-yield topic in the Biological and Biochemical Foundations of Living Systems section of the MCAT, and it shows up everywhere. Experimental passages, lab-based figures, and questions that ask you to interpret results from electrophoresis, ELISA, or mass spectrometry. Today we're breaking down the core techniques the MCAT expects you to recognize instantly. Not the deep biochemistry behind them, just what each method does, how it works, and how it might appear in a passage. If you understand the purpose of each technique and the kind of data it produces, questions that once felt confusing become straightforward pattern recognition. That's exactly why protein analysis is such a powerful topic to master for a higher MCAT score. Protein analysis, studying structure and function. Protein analysis is all about understanding a protein's properties, its size, separation pattern, structure, abundance, and interactions. Most MCAT questions don't ask you to perform the technique, they ask whether you can interpret what the results mean. Electrophoresis, separating proteins. Electrophoresis separates proteins by charge, size, or both. The most common MCAT-tested version is SDS-PAGE, which denatures proteins and coats them with a uniform negative charge. That means proteins separate almost entirely by molecular weight. Smaller proteins travel farther down the gel, a detail MCAT passages often test. Western blotting, detecting a specific protein. A Western blot starts with SDS-PAGE separation, then transfers the proteins to a membrane. Antibodies bind to the protein of interest, allowing you to visualize just that one protein out of an entire mixture. When a passage shows bands that increase or decrease under certain conditions, it's almost always a Western blot. Enzyme-linked immunosorbent assay, measuring protein levels. Enzyme-linked immunosorbent assay quantifies how much of a specific protein is present in a sample. Antibodies capture the protein, and a color change or fluorescence tells you the protein's concentration. This technique appears frequently in immunology-based passages or experiments measuring hormone levels. Mass spectrometry, determining protein, mass and sequence. Mass spectrometry ionizes protein fragments and measures mass-to-charge ratios to identify amino acid sequences or confirm the identity of a protein. If a passage mentions peptide fragmentation or mass-to-charge peaks, it's pointing to mass spec. X-ray crystallography and NMR, determining 3D structure. X-ray crystallography determines protein structure by analyzing the diffraction patterns created when X-rays hit a protein crystal. This gives atomic-level resolution. NMR spectroscopy provides structural information by analyzing how atomic nuclei behave in a magnetic field. On the MCAT, these techniques signal that the question is about structure, shape, or binding orientation. Circular dichroism, CD, studying secondary structure. CD spectroscopy measures differences in how left and right circularly polarized light are absorbed by a protein. The patterns indicate whether a protein has more alpha helices, beta sheets, or random coil structures. It's a classic technique for secondary structure analysis, especially in folding experiments. Proteomics, studying all proteins at once. Proteomics examines the full protein content of a cell or tissue under specific conditions. It identifies and quantifies hundreds or thousands of proteins at a time, giving insight into biological pathways. If the passage discusses global protein expression, think proteomics. Protein-protein interactions, understanding complexes. Protein interactions are studied using methods like co-immunoprecipitation, yeast-2 hybrid, and pull-down assays. These techniques reveal whether two proteins bind to each other, a detail often tied to signaling pathways or gene regulation. When a passage mentions binding partners, it's testing interaction techniques. Mastering protein analysis gives you a huge advantage on MCAT experimental passages. When you recognize the technique being used, the entire figure makes sense. The gel bands, the peaks, the color changes, the structural diagrams. And once you understand the purpose behind each method, the questions become predictable and easy to reason through. With that said, here are just a few of our student success stories, students who improved their scores and even got into medical school. If you're preparing for the MCAT, click the link below to get started with your own preparation. We specialize in helping students reach a 515+ score guarantee, and we'd love to help you too. And don't forget, Subscribe to our channel for more MCAT breakdowns like this, and follow us on Instagram for extra tips and resources.